Okay, folks, let's get going here. I'm just gonna go and mention, I'm not recovering all the material now. I'm just telling you the areas that you need to look at. Three parts to this. One is what the membrane does. Second unit is going to be based upon energy itself, its nature. And the third then is safe restoration that we have just talked about. You do need to know a little bit about the permeability of membranes. You should know things such as what is diffusion, what is osmosis, what is an empathic molecule. You should, once again, you've already had to do this, but be able to draw that lipid membrane. The lipid bilayer, phosphates on the water side, lipids internal. Also, now with the membrane, you have to understand there will be proteins that are involved with that membrane surface. One of the big things about a membrane is its fluidity, which means its ability to move. Remember, we can have motion within the membrane, movement of the actual molecules, fatty acids. They can go side to side and just exchange places, or they can go from one end to the other end. The most common is the lateral movement. That occurs fairly frequently, if you notice. 10 to the 7 times per second in molecules. So these molecules are always kind of moving around, changing places. That's the fluidity. Now, this is gives you an idea of the membrane parts, and it shows you some of the proteins. Remember, these right here, the little purple one that you see up there, with the green coming out, that purple represents a protein. The green represents carbohydrate. Whenever you put those two together, they are called a glycoprotein. They are often the ones that act as receptors on the cell to communicate with the cell. Those are often the ones your body's blood cells look over to determine are they yours or not. Yeah. And glycoprotein. Look at it right up here. It's got the names of those right there. That is a glycoprotein. If it was on over here, sometimes they're attached to a lipid and they're called glycolipids. So it's just glyco is referring to carbohydrate, and then the proteins, protein, lipids, lipid. Note these different kinds of proteins. We have peripheral proteins that are on the inside part of the membrane. Then you have integral proteins that go through the membrane. These proteins are important because they are the ones that allow movement in and out of the cell. They have channels that can open up and let molecules in or out, but it's a control method. Also, that's important to remember, in the membrane, to help with fluidity, to help keep it fluid, particularly when there's cold temperatures, there are little molecules of cholesterol. They help to prevent packing from within those lipid tails. This tells you about that, and also don't forget, if you've got membranes, just as we've said, fatty acids, saturated or unsaturated, most of the membrane are going to be unsaturated. They're going to have the ability to move more. A saturated membrane in cold temperature, for example, would just basically pack up and just lose its fluidity. shows the unsaturated. And those that are unsaturated take up more space than those that are not, than are saturated. Cholesterol, as we said, plays its part. Know this definition of a peripheral protein and an integral protein. Integral proteins go all the way through the membrane, so they have to have a part that comes in contact with water on the outside of the membrane. That has to be hydrophilic. The part that goes down through the fat layer, the lipids, fatty acid, has to be hydrophobic. So you've got polar out near the water, non-polar in with fatty acid chains. Those are hydrophobic regions. And proteins, remember, can have some parts that are polar, some that are non-polar. Those fatty acids or amino acids come together to make that. This is just a picture showing there are going to be polar areas and then non-polar areas that go through the fat.
So the C terminus and the N terminus would be polar?
That's just on the end of the proteins, yeah.
Right, so they would be polar and hydrophilic, while the middles would be non-polar and hydrophobic?
Yeah, pretty much. The middle can be whatever's needed for the situation, which it can be, in this case, it's non-polar. In other words, it likes fats. Polar likes water. Non-polar would be fatty. You do need to know these functions of the membrane. I'm not going over them all, but it gives you 6 functions of those membrane proteins. So know those. This is just pictures of those. One of the big things is cell-to-cell recognition. Carbohydrates on those proteins play a huge role in cell-to-cell recognition or making receptors.
In the book, for the 6 functions of membrane, they have like a definition for each of those. Do we need to know that, or should we just know the functions themselves?
Fine.
Thank you.
Remember this part. We've covered this before, but you probably get a question on it. The distribution of the proteins, lipids, and associated hydrocarbons or carbohydrates in the plasma membrane is determined when the membrane is built by the ER and the Golgi. They work together. ER actually makes it, the Golgi transports it to where it's supposed to be. So if you have the question, where is the membrane built, you really should answer, you can answer both, ER and the Golgi. This shows some of these proteins as the proteins are being made. Here's the ER. It's making these proteins now in membranes, and now you can see that will start to attach these parts to the receptors. This is the Golgi apparatus, which will then move them out to the membrane and either release them out of the cell or join the cell. So the ER makes these membranes and proteins. The Golgi transports them. Selective permeability is, if you read it, is just the ability of the cell to decide what comes in and what goes out. There are different ways that this can happen. This is once again just talking about the hydrophobic and hydrophilic part of the membrane and the molecules that go through. A transport protein allows substances in or out. For water, there are proteins called aquaporins that allow water to come another type of protein is called a carrier, where it actually binds to the molecule, changes the molecule's shape so that it can go through the channels. Diffusion is just the natural tendency for water to spread out evenly. That is diffusion, yeah.
Okay, so the carrier protein, does it change so when it binds to the molecule, does its shape change, or does the shape of the molecule it binds to?
Everything changes when combined. Notice substances diffuse down a concentration gradient from more concentrated to less. The diffusion of a substance here across these membranes is called passive transport. There's no energy, nothing is helping it. Osmosis is this kind of diffusion. It is just natural characteristic of water to want to even out, so it will go across the membrane. But notice here, water diffuses across the membrane from the region of the lower solute concentration to the region of higher solute concentration in an effort to dilute it so it becomes equal. So water moves from low concentration to high. This is another example. You do need to know these terms, what they mean. Tonicity, isotonic, hypertonic, and hypotonic. I've got to move along a little bit here, so I don't have time to go over all of them. Know basically what they are. Know which way water moves. Like in a hypertonic solution, solute concentration is greater outside than inside, so water goes from the inside of the cell to the greater concentration, which is outside, so the cell would shrivel up. Hypotonicity, water concentration is greater inside the cell than out, so water moves in, and you can have the cell to actually implode, and that's what it shows you here.
What if it says that a cell is hypertonic to its environment? Does that mean that the water flows in? It's in a hypertonic solution?
If it's in a hypotonic solution, right there.
So the cell is
That means
hypertonic.
If it's in hypotonic solution, the concentration is lower outside the cell. Water moves in, causes the cell to rupture. If it's in a hypertonic solution, the lower concentration is inside the cell, it moves out and shrivels.
The reason I ask is because one of the examples was a paramecium, and it said that it was hypertonic to the pond
Amen.
water.
Guys, go ahead.
It said that the paramecium was hypertonic to the pond water, and I know it has a contractile vacuole, but I was wondering if does that mean that the water is flowing inward because it has more solute inside, so the water is.
Paramecium have a vacuole. They control their own water input pressure.
Right, so if it says it's hypertonic to the water, does that mean that the water outside of it's trying to flow inward? Okay, thank you.
Osmoregulation. It says right here, the Rhodus paramecium is hypertonic to its pond water environment. It has the vacuole that can pump water out or in. Osmoregulation is just the control of the solute concentration. That's important to all organisms to be able to control the quantity of electrolytes, whatever. That is called osmoregulation. There's the little vacuole in the paramecium. It can control its own internal environment. Know what turgid and flaccid is, like for plant cells, whenever the water leaves, if it's in a go back to this part right here. If it's in a hypertonic solution, which means water leaves the plant cell, if it comes out, this membrane pulls away the peptide. That's called plasmolysis, so remember that. Facilitated diffusion is these proteins help to move things across. They facilitate the movement. Same with ion channels and so forth. Note this, active transport moves substances against the concentration gradient. So there's always going to be situations where a solution outside, for example, could be in a hypertonic solution, but you can get them, instead of going from low to high movement, high to low. Remember, a solute will want to move to a lower concentration to try to even it out. Water tries to help with that by moving from the low concentration to the more diffuse to thin it out. Not like that. So when this occurs, when a substance is moved against its gradient, that takes energy, ATP, and that is called active transport, done by, as you see, the proteins. Sodium potassium pump is an example of how, whenever you want to get an electrical charge, for example, across the membrane, you can pump the positives out on one side, make the electrical gradient have a jump over to get a charge. Acid transport, they just come in on their own. Here's diffusion. Sometimes a protein can act as a specific channel, so a specific shape of molecule can come through it, no others. Then there are some that actually use energy to bring it across. These are both passive on the left here. They do not use energy. Active does. This talks about these gradients again. This one's electrochemical. That just simply controls where the positive and negative ions are on the membrane. And we just looked at ATP synthase, where we had hydrogen goes on one side of the membrane, carried by these proteins that we talked about. It's a little bit different than this, but it does the same thing. It separates the charges.
Here, is it showing that ATP is actually being used to pump across the membrane?
Here it is.
Okay, but in the ATP synthase case, it's actually produced from the exergonic
ATP
fluid.
synthase is different. The hydrogens are put on the membrane by other proteins. Here, the hydrogens are just being pumped out to that across the membrane. That's what a proton pump does. It does take energy. ATP synthase does not. Other proteins are carrying. That's basically what I wanted you to know about this chapter. You don't have to get into endocytosis and so forth. Unit 8 on energy. Remember, metabolism is the sum of all of the organism's reactions. A metabolic pathway is taking something like glucose, breaking it down, metabolizing it. It has a specific pathway that we looked at. Also remember, anytime anything is catalyzed, enzymes are required to do that. Should remember, cellular respiration is an example of a catabolic pathway. Photosynthesis would be an example of an anabolic pathway. Anabolic pathways make things, catabolic pathways break them down. Catabolic gives off energy, anabolic uses energy. And look at the definition of energy, get the ability to do work. You should know here these things, what kinetic energy is, what potential is, what chemical is. Remember, chemical energy, energy stored in a bond. It is potential energy. It's stored in a bond. If there's a reaction going on, then it would become kinetic energy. Heat is going to be a measurement of thermal energy that is produced when we have reactions. Thermodynamics, study of energy of transformations. The first law is the big one. It says we cannot create or destroy energy.
Do we need to know all three or
You need to know this one. The second law is the one about entropy. That means, basically what it means is with us is there are no reactions that are 100% efficient. We always lose, so that goes towards disorder. Those are the basic ones there. That's where we're talking about order and disorder going towards in the systems. Entropy, remember, is moving towards disorder. Free energy, just remember, is the energy that is available to be used by an organism. There's a lot of energy in molecules, but not all are available for us to use. Hopefully everybody knows what equilibrium is, which is just a state of stability. Obviously, no exergonic and endergonic. Exergonic giving off, endergonic using energy. We've already mentioned catabolic pathways release energy, anabolic uses energy. Do know the three kinds of work that a cell does: chemical, transport, mechanical. ATP is the energy shuttle or carrier or the energy taxi that is going to take energy, it's going to capture energy and then take it from one place to another. Note what it's made up of. It's going to be a nitrogen base, sugar, ribose, 3 phosphate groups. Should be able to draw it again. When you draw it this time, last time we just had adenosine. Adenosine is when you have a base and a sugar combined. So you can draw it that way. You can draw one block. It says adenosine, which includes both the sugar and the base, and then the 3 phosphates. And you can draw the phosphates, as we said before, just a circle with a P.
So we can draw it exactly how we drew it last time? We don't have to break it into adenine and ribose?
Nope.
Okay, thank you.
You need to know that it's there, but you don't have to draw it that way. And hopefully everybody knows that energy is going to be released whenever that last, the terminal phosphate, is released from ATP. That release of that phosphate releases the bonding energy that was required to attach it to ATP. Whenever you add a phosphate to another molecule, that is called phosphorylation. Phosphates are in the cell and available and ready whenever it is needed. Remember, catalysts, they speed up a chemical reaction in chemistry. They are called catalysts. Enzymes are in organisms and are protein catalysts. They speed up the reactions in our body. Without the enzymes, very few of the reactions would occur fast enough to support life. Also, don't forget, almost every single thing involves the use of an enzyme. Know what activation energy is? Know how enzymes work by lowering activation energy. Know the base parts of an enzyme. Enzymes act upon substrates. What kind of molecule is an enzyme? A protein. So whenever that enzyme combines with a substrate, what it's going to work on is called the enzyme substrate complex. Somewhere on that enzyme is going to be an active spot where that substrate will bond to. Induced fit can bring chemical groups together. In other words, that enzyme's got one position whenever it gets the substrate attached to it, the shape can change. Notice how this part right here, up in here, closes to capture then the substrate. So it holds it into position and closer than for having the reaction occur. You don't need to go through that there. Just understand how the enzyme works. You've got your substrates up through here. They go to that active site of the enzyme. The reaction takes place. The enzyme then releases the new products. Remember this, and you can pretty much be assured that you're going to be asked this. An enzyme does not become part of the reaction. It only makes it occur. It does not become part of the product. There is no enzymes consumed. It will, the same amount of enzymes going in will do its job, and that will be the same amount of enzymes that you have left over. What can affect an enzyme's activity? It's protein. So temperature, pH, or chemicals can all affect the way an enzyme works. There are ideal or optimal pHs, temperatures for different enzymes. They're not all the same. Each enzyme can have optimal conditions. For an enzyme to work, they have little helpers called cofactors. Cofactors can be inorganic, such as just an element, lithium, or it can be a molecule, such as a vitamin. That is called a cofactor. Yeah.
So NAD plus would be a cofactor?
Yep.
Okay.
Competitive inhibitors bind to the site, active site, to prevent the substrate from binding. Non-competitive change the shape of the enzyme so it will not be able to receive the substrate. Normal, competitive, something comes in and blocks the substrate. In non-competitive, it actually changes the shape of the enzyme so the substrate cannot come to it. And you can often do that by changing. This would be where the cofactors would often come to. So if you put in a different cofactor that will bond to that site, it can block it. And remember too, cells are just like an engine. They need a certain amount of fuel, but you cannot give it too much or it will not work. It needs to have the correct amount of products produced, correct amount of resources to substrates. So the cell has the ability to control, like with these proteins, that can control how much that comes in or goes out. Allosteric regulation. The molecule can bind at a protein at one place and affect its function like at the active site. Such as that, or non-competitive inhibition. Most of these are going to be peptides, polypeptides. Didn't ask you about cooperativity. There's other examples, we didn't get into it that much. Feedback, just remember, producing the product that the enzyme is supposed to produce, once it's present, acts as an inhibitor. The enzyme only works when it doesn't have enough product. When it does have enough product, then that product shuts it down. Whenever it drops too low again, then it starts again. So in feedback inhibition, product is only being made when it's not there. In other words, it doesn't just start making it, keep making it. Once you get a certain amount, it cuts it off. We didn't really talk very much about where they're located because they're basically everywhere. And they do a lot of things. Notice it says sometimes they're going to be in organelles. They can be located in mitochondria. They can be located about everywhere. Now, on the chapter that we just covered, we're going to go through quick. You want to know what is the role of oxygen in respiration. You want to know where the energy comes from, which is the sun. You need to know that there's two basic ways, two pathways primarily to get energy. And energy, when it's released, is catabolic reaction. Fermentation, which does not produce enough energy for a large organism to survive on. That's mainly going to be smaller organisms. And aerobic respiration uses oxygen. There are some organisms, certain bacteria, in fact, that require no oxygen in their environment. They go through anaerobic respiration. They can get out basically just about as much energy out of a molecule as we can through aerobic, but it is done in a different pathway. It's not going through glycolysis and things like that. And these are bacteria who are themselves little energy machines. You know, their whole makeup. Yeah.
So, if we're asked what are the two pathways that release energy, basically, that would be fermentation and cellular respiration?
Fermentation, aerobic respiration, and anaerobic.
So would that in total be cellular respiration, or we would specify aerobic respiration and anaerobic?
Aerobic and anaerobic are two different things. They have to be treated differently. Anaerobic is primarily in bacteria. Cellular respiration is a combination of aerobic and anaerobic. Cellular respiration is what most higher organisms go through. Lower organisms will go, many of them strictly anaerobic. That's why at the end we run things like obligate anaerobic. They cannot survive in the presence of oxygen. Facultative can. They can use oxygen or not. They can still go through respiration. We're talking about cellular. As it says, includes both aerobic and anaerobic as the types. Should know the equation for cellular respiration, which is the opposite of the one we learned last time. Glucose and oxygen gives carbon dioxide and water. You also should make sure to remember The two waste products for the metabolism of a glucose molecule, the waste products will be carbon dioxide and water plus energy. No redox reactions and oxidation or reduction actions. Oxidation, electrons are given up from a molecule. In reduction, a molecule gains electrons. So if they've gained an electron, they are said to be reduced. If they give up an electron, they are said to be oxidized. And notice what this says here as an example. Glucose is going to be oxidized. It's going to be giving up electrons. Oxygen is reduced. What is oxygen reduced to? Water, carbon dioxide. That right there tells you basically why we need oxygen. We're going to be pulling off electrons. Things are going to become oxidized. What's going to pick up those electrons or be reduced is going to be oxygen, and it will make, as a result, carbon dioxide and water at the end, both of which are non-toxic. And one of the reasons why your body wants to use those for energy. No, NAD positive. Another thing can give up an electron and then reduce NAD positive. The electron's going to be in the form of this hydrogen proton and electron. So NAD positive becomes NADH. Get ahead a little bit, that's what we're already talking about. Here are the three stages you need to know. You need to know what important things happen in those stages. Most important products for glycolysis, it's pyruvate. Citric acid will be electron carriers, NADH and FADH. The most important thing to come out of oxidative phosphorylation is ATP, energy. So out of glucose, pyruvate, citric acid cycle, electron carriers, oxidative phosphorylation is then ATP. Know where they happen. Glycolysis in the metabolism and what the products are it combines with to go across the membrane, giving up carbon dioxide to make the acetyl group. Then the products, acetyl group goes into the citric acid cycle. There's going to be NADH made and FADH that will then go into the transport cycle. There will be 4 molecules of ATP made in glycolysis. 2 must be consumed to drive the reaction, so there is a net gain of 2. And there will be 2 made in the citric acid cycle. Oxidative phosphorylation occurs because an electron is given up and used then to phosphorylate ADP. Substrate phosphorylation is an enzyme, does what an enzyme does. It acts as a substrate holder, and it brings 2 things together in which the phosphorylation can occur. It reduces the amount of energy that needs to make that reaction happen. Then something is oxidized, gives up energy, and then phosphorylation occurs. That's called substrate level phosphorylation, and remember, oxidative phosphorylation occurs only in the electron transport system. Substrate phosphorylation occurs in glycolysis and the citric acid cycle. Yeah.
So, so substrate phosphorylation is basically basically enzymatic activity, basically, right? I'm sorry, the substrate phosphorylation.
Yes.
Okay, and then oxidative phosphorylation is just the electron transport system because we're dealing with giving up electrons. Okay, that makes sense.
Oxidizing.
Oxidizing.
Other things being reduced. This is a summary of what happens in glycolysis. Remember, 4 made, but 2 will be used, so there's going to be a net only of 2. You need to know this investment phase. You need to know the product and the enzymes. You do not have to know that for the payoff phase, but you do need to know where energy is used, steps 1 and 3, where it is given off, 6, 7, and 10. You need to understand how pyruvate gets across the membranes. We've already talked about making a carbon dioxide, combining with coenzyme A to make acetyl-CoA. Once it goes across the membrane by the help of protein carriers, it will release the CoA. The acetyl group will then combine with oxaloacetic acid or oxaloacetate to make citrate. Then we start to pull the electrons and protons off of citric acid. Citric acid, then, this shows the process, should be able to draw this formula right here for the acetyl group. The carbon, double bonded to an oxygen, bonded, single bond to another carbon, and then to 3 hydrogens. Yeah,
So the acetyl group, not the pyruvate?
the acetyl group.
Okay, thank you.
The acetyl group, note, is 2 carbon. It will combine with the 4 carbon oxaloacetate. And then we have regenerated citric acid. 2 carbon, 4 carbon. Yeah, so what, those all in.
Oh, I actually do. Do we need to remember the carbon, the number of carbon changes that occur? I mean, when it changes.
Remember
2 to 4.
what I told you right there. 2 carbons in the acetyl group to make 4, matched with oxaloacetate to make 6 carbon citrate. You do need to know the products. You do not have to know the enzymes. But you need to know in step 1, we've got citrate. Step 2, iso citrate. Step 3, alpha ketoglutarate. Step 4, succinyl-CoA. Step 5, succinate. Step 6, fumarate. 7, malate. And 8, oxaloacetate.
So we need to be able to identify all of those.
You should put them in order, yes. And you should know it, what is regenerated in the citric acid cycle. Oxaloacetate is regenerated. So basically what is happening here, that acetyl group is being metabolized and broken down, and it's going to become either carbon dioxide, as you see in the little gray things, or water. And when that happens, at the end, all of that's broken down. We're back with what we started with, which is oxaloacetate, which is ready to receive another acetyl group up here. When you look at this part right here, remember the acetyl group is in the white. This brown part up here represents the CoA, which is just a carrier. It does not become part of the molecule. It carries the acetyl group to this site, and it leaves, and it will come back in down here again to do the same thing to help the succinyl group move over to become succinate. Go ahead.
The SH was, did that represent succinyl? I noticed that it was, when it first binded to, so
Succinyl-CoA.
So up there, when it says CoA, and then it says SH-CoA, what is the S representing?
That's a carrier
Oh,
molecule.
it's ionized or non-ionized?
Yeah, we gotta move along here, we've only got 1 minute. This is oxidative phosphorylation. No, it's 2 parts. The electron transport system and chemiosmosis. Note that the electron transport system is going to carry those electrons. Carriers are going to come into the electron transport chain, release the electron. The electrons are going to be carried down this chain that we see right here. They will stay within the inside of the membrane, come back to. The hydrogen is then going to travel through these proteins, such as we saw in proton pumps. They are going to be pushed through. That does require energy, these proton pumps, to move it on the other side of the membrane. So that is a form of active transport there when we take those protons and push them out. Remember, they're going against the gradient, so it takes energy. Eventually, they will accumulate and cause another electrical gradient or chemical gradient on the outside of the membrane. The hydrogens will be pulled back down through ATP synthase, then reunite with their electron, which has been captured by oxygen to make water. The hydrogen gradient is called the proton motive force. Remember, 34% of the energy in a glucose molecule is taken out. Only 34%. There'll be a net extraction or making a net of 32 ATP. You do need to know there's 2 that comes out of glycolysis, 2 that comes out of the citric acid cycle, and about 28, and whether it's 26 or 28 is referring to what kind of cell or organism that it is. You need to know about fermentation, the 2 kinds, alcohol and or lactic acid. One of the things you should know here, we've got that acetyl group. This whole thing is pyruvate up in through here. But we're going to get rid of the carbon dioxide to get down to the acetyl group, which is then going to be attached, have a hydrogen attached to that end, become acetaldehyde. And then it will turn into alcohol, ethanol. Notice what happens if we don't have oxygen. We have to go into fermentation, lactic acid fermentation within the muscles. This is a summary of what's going on. It's not really anything different. You do need to know what an obligate anaerobe is. You need to know what a facultative anaerobe is. To understand a little bit about how fats come into the pathway right here. Fatty acids got to be held together by a glycerol molecule and a fatty acid chain. They, as we will see here, glycerol will go into glyceraldehyde. Fatty acids can be turned directly into acetyl-CoA. So you could be asked a question, how do fatty acids come through respiration? You would just say, they don't really go through glycolysis, as you said. Glycerol goes into glycolysis, but fatty acids just become acetyl-CoA.
So we would say that they go through the citric acid cycle?
They're turned into acetyl-CoA that goes directly into the citric acid cycle. They do not go through glycolysis.